Thank you very much for the invitation. Great pleasure to be here. Big fan of this webinar seminar, which also makes me helps me to stay up to date with what's happening in this space. So I'm going to talk today about quality criteria for degraders. I guess we are all in this call very excited about this new modality, small molecule degraders. However, when we are coming from drug discovery, it's also important to realize, and I'm doing this here, but just showing something one of my Twitter buddies posted recently from Keith Hornberger, that even with the greatest tech, in the end for drug discovery, the question is, how do we actually select the right target to work on? And I think this also fits nicely with something Bill Kaelin in this webinar seminar 2 weeks ago has discussed. And he also made the point that drug development success is closely linked to high quality targets. So essentially being able to validate a target with genetic means or other means to then turn this into successful drugs. However, in drug discovery, we also need to realize that target discovery is one of those spaces which has been hampered also by low reproducibility. And I'm citing here a study from my former colleagues at Bayer. It's 10 years ago, but still, where they could show that for about 2/3 of their internally run projects, they were running into significant issues with reproducing some of the published data out there. And this is not only true for Bayer, it's also true for Amgen. They were even, let's say, less successful in confirming what they were reading in the literature. And there has been then a study or a paper looking at the economics impact of this low reproducibility, and it's really mind-boggling the numbers you can come up, essentially money invested into drug discovery and development, which probably is lost based on the fact that just the underlying hypothesis is already wrong. For a chemist or in general, if you think about what may be the underlying reasons for this low reproducibility, I think it's safe to say that probably low quality tools are one part of the story. And these tools can be actually small molecules used in these studies. So sometimes for chemists, it's easy. You just look at the structure and then you can already make up your mind. But obviously in the broader community, which is very interdisciplinary, it's not that easy. And I'm giving you here one example, which is quite well known, curcumine, excellent paper from about 7 years ago in JMed Chem. And in a nutshell, this compound was claimed or is still claimed to have a long list of potential positive effects. If you're still in grad school and you're planning your spring break, one could say that whatever health risk you may be facing during spring break, if you believe what you can read in publications, this compound is probably the way to overcome these health risks. However, in reality, and again, for the chemists on the call, looking at the structure or being a bit familiar with this field, it doesn't come as a big surprise that obviously this is not true. If you ingest this compound, the first challenge already is that you do not really know what you ingest because it's usually impure. If you want to go for spring break, you need to be aware that the compound is actually not stable in sunlight. You don't have to worry too much about this drug becoming wet because it's essentially not soluble in water, which already raises the question what we are actually looking at. In a nutshell, there have never been placebo-controlled or blinded relevant clinical studies showing any benefit of this compound. Obviously, this is a relatively easy to spot low quality compound, and it has been known for quite some time, and there have been efforts also to make the community aware that this is obviously a compound which should not be used to validate targets. However, it requires really a community effort to uphold these standards because in reality, you can publish this. You can publish this in Nature, but this doesn't mean that from there on, this compound is not used anymore for exactly that reason for target validation. To just give you one example, which I picked because it was published about a year later in a quite, I would say, high-ranking journal. I'm focusing here very much on small molecules. I think it's clear that most of what I say apply to other tools as well. I want to highlight one very recent paper which looked at quality of publicly available or published antibodies. And there you can find very similar findings. And obviously, this is at least as relevant for target discovery, reproducibility. Another aspect one always needs to keep in mind when one puts such compounds into the public domain is that when you poison the literature with a compound, it doesn't go away. So you can publish a lot about why this compound is maybe not fit to be used. It only takes a pandemic, and what then happens is that all these wonder drugs, and curcumin is just one example, pop back up and are claimed again to be the solutions to all kinds of issues, for example, in a situation of a global pandemic. And you can just guess again how much in money, discovery money, then potentially goes into such studies. There have been very important activities for close to 2 decades now in public-private partnerships to propagate quality criteria for chemical probes. And one of the most important one of those has been the Structured Genomics Consortium. And I'm going to use now a bit of time to go through a set of criteria the SGC has been using, optimizing over quite a long time, and has been now successfully applied to a significant number of public domain chemical probes. And this is important to then discuss what elements of these quality criteria should also be applied to degraders. So from a big picture, these criteria fall into 2 categories. One category is the target-related criteria side. So what level of potency would you want to see in a high-quality probe? And here it's especially important to highlight that the clear recommendation is to have cellular potency below 1 micromolar. And one of the rationales behind it is you want to make sure that you can measure your desired pharmacological effect and differentiate from unspecific, for example, cellular toxicity. Small molecules, you can usually use then up to 10, 20 micromolar, then solubility will limit what you are seeing. So with an activity in the sub-micromolar range, you still have a nice split factor to differentiate, to check at the higher concentrations how specific is the effect you are seeing. Selectivity 30-fold within the family is a recommendation. And a second really important element is to show that you are actually directly engage the target, for example, by biophysical means. The second part of the quality criteria are more in the chemical matter category. I guess this part is relatively easy for chemists. So the recommendation is check for obvious elements in your molecule which may lead to promiscuous unspecific activity. A point I really want to emphasize, and I will come back to that, is the availability of a control compound. This would be a compound from the same chemical series, but significantly reduced or no activity on the target of interest. And it always helps if such probes have sufficient solubility, and obviously they should be stable under the typical assay conditions. So this is a high-level set of quality criteria, and we will come back to the individual aspects on this slide when we discuss degraders. There's a significant number of such high-quality probes in the public domain, and I'm just highlighting here a few of the good sources: Probe Miner, the Chemical Probes portal, and Boehringer Ingelheim's OpenMe. And there are 2 examples here. If you want to go a bit more into the details to activities I've been involved in the past, so in epigenetics back in the Bayer days, is my 2 inhibitor. And more recently, and this is, I think, quite important, kinase inhibitor. There are now more and more highly selective kinase inhibitors in the public domain who allow to study the biology of so far understudied kinases, like this family of kinases which is involved in modulation of splicing. Now, you can put high quality probes into the public domain. The question is, what does the community then do with these probes? And there was a really interesting study by Lenka Munoz published last year, where they looked at a number of these high quality probes and then collected, published studies and checked how are people actually using those probes. And what was, I think, a very a positive finding is that in about 80% of the cases, the studies actually comply to, for example, recommended concentration ranges. So with these probes, there's always a recommendation how to use them. And most of these studies, although there are some outliers, are using these recommendations. What is really, I think, I have to say I was a bit speechless, is that at the same time, essentially no one is using the available negative controls. So there are a number here in this table of compounds which come with a matched control compound, and essentially nobody is using these control compounds, which I think is interesting. If you would think about using a short hairpin RNA or an antibody, it's kind of a given that you use controls, but for small molecules, it's not. So this is clearly something which needs further community efforts to really drive home this point, how important it is to do the controls. What we have done is we have joined forces with a few colleagues from academic and industry background from the Chemistry and Cancer Research Working Group and published recently a high-level recommendation, best practice, experience, how to select and use these small molecule probes in cancer discovery. And we published it in a paper in a journal which on purpose is targeting the community who is doing these studies, because we feel it's also about bringing this message to the right community. And when we were doing this, we realized, what about these new modalities of small molecule drugs which are now emerging? And I can be very quick on this one. In this audience, I think everybody here knows that small molecule drugs have had and still have significant impact in the clinics, but that there are situations where you actually want to completely remove the drug, and there are ways how you can do knockdowns, but from a clinical perspective, the traditional ways, or traditional, so the biological ways of doing it are relatively complex and also cost intensive, which means that the small molecule degraders really sit at this sweet spot between these both worlds. If we are now more interested in discovering new targets, uncovering biology, it's a bit almost the other way around. How do we typically identify new targets? By doing knockdowns with genetic ways. And then there is this challenge, how do we translate this into a drug? While using small molecules, and I have discussed this already, selectivity can be challenging, and also the number of targets you can go after. So also when you're more interested in target discovery, degraders really sit at this sweet spot with a lot of potential to discover new biology, but obviously, again, quality matters. One example to maybe explain this point even better is, if we would be all now in a room, I would ask you, would you consider lenalidomide as a high-quality chemical probe? And then I would ask people to raise their hand and see what is the feeling. And I think most of you know that lenalidomide is a very successful drug. And I'm just giving you one example here for clinical impact, survival data, and this is really huge. So I think there's no doubt that this is a compound which is really making a difference for patients. But is it also a good chemical probe if you want to study biology? And again, in this call, probably everybody knows how lenalidomide works. It binds to cereblon, creates a neosurface, and then binds a whole family of G-loop containing proteins and degrades many of them. So it's polypharmacological by nature, which means lenalidomide is an excellent drug, but a very bad chemical probe if you would, for example, want to study biology of, let's say, IKZF1. Because you will always see this mixture of cellular effects. So it's important to understand this difference between what is an excellent drug and what is an excellent chemical probe. Now let's go back to these degrader criteria, or this general probe criteria, which I had introduced, and convert them into something we can directly apply to degraders. So again, we start with potency. As I had made this point before, the recommendation is to see degradation as the DC50 in concentrations below 1 micromolar, to be able to see a split to general cytotoxicity, preferably in the same cell line. Disclose relevant concentrations, how to use your probe. So it's important, if you have an hook onset concentration, report that. There is then a kind of a discussion, how deep do you want to go? And the clear recommendation is go for as complete degradation as you can. And why is this important? And here I just give you one, I think, really important study from the literature, from the Linden lab, where they generated PROTACs for Aurora kinase A. And what they found is that although they were able to generate potent PROTACs, they didn't see the phenotype they were expecting. And when they looked more closely, they realized that they are degrading a large fraction of Aurora, but not that part which is sitting in tight protein complexes, specifically at the centrosomes, and that essentially the biologically relevant fraction of Aurora they didn't touch. So that's why I think it's really important to be aware, degrade as much and as completely as possible, and be a bit careful when you have incomplete degradation when interpreting phenotypes. I made a point that's important to show target engagement. For the degraders, it's important to understand the mode of action. So do the controls, is the degradation modulated via the expected E3? It is important to understand which E3 is actually behind the activity, because engaging E3s, and you can see some examples here, can by itself contribute to the phenotype you're observing. For many of the recently used E3s, there's not so much known. There's a great webpage from the Han lab, which provides a lot of information. But again, there obviously for biology, there are many questions. And just that somebody, let's say, or you try to target a certain E3 ligase doesn't mean that the activity is actually mediated by this E3 ligase, as this elegant recent study has shown. So it's really about have you done the appropriate controls to really understand what your compound is doing. Selectivity, this is maybe expected to be the easiest one. So obviously you want to assess close neighbors. But then the question really starts, how can you really completely assess the selectivity of a degrader? And I think the iceberg picture is a really good one. Obviously, you want to do an unbiased proteomics study. And this would help you to really understand, and this is kind of what's on top of the waterline here, what other proteins are degraded. But this is also a situation where you need to be aware there's a lot under the waterline. There are these, let's say, known unknowns, proteins which are not expressed in the cell line you're looking at. So the question is, how many cell lines do you have to study? What is, I think, heavily underemphasized when talking about selectivity of degraders is what else is your bifunctional degrader or glue degrader actually doing? If it's binding to E3s, what else is it binding? If it's a covalent engager, and keep in mind, it may engage other proteins but not degrade them. But by engaging other proteins, you may already also generate phenotypes by inhibition, by changing localization, by changing dynamics. So there is a lot which can happen here, which you will not be able to see in the proteomics studies. And that's why I think these efforts to generate ways how you can look at the biointeractome are really important also in this space when you want to understand what a degrader is actually doing. The chemistry side of things is, I would say, relatively straightforward. The same applies to panes, promiscuous groups. If you have covalent E3 engagers, obviously you want to understand what else you're binding to. Yes, as for other probes, provide a control, use a control. For a degrader probe for an E3 engaging compound, it's important to look at the non-E3 binding controls. This can be modulation or let's say methylation of a cereblon binder, inversion of the VHL binder. So essentially a related compound which is not engaging the E3 to differentiate what is happening beyond, for example, degradation. What I am a big fan of on top, especially if one works with cereblon-based PROTACs, is to use these capped E3 binders. So something which is closely related to the cereblon engager, which has part of the linker as a control to get a feeling of what am I actually doing to the neosubstrate space of cereblon with something which is closely related to my degrader molecule. And finally, as for typical probes as well, you want to have solubility, you want to understand stability. I think everybody who has worked with cereblon engagers is aware of the chemical instabilities, especially in the thalidomide type system. So hydrolytic instability, but also that the stereochemical integrity is often not there. It's sometimes not so easy to predict how linker structures influence those activities or instabilities, but there are now a number of excellent studies, for example, here from the groups in Bonn around Christian Steinebach, disclosing lots of these data to help predict what makes sense and what makes no sense. More personal comment from my side, if you want to use these degraders in vivo, I would always recommend stay away from the PEG linker-based PROTACs. If you have a PEG linker in between, this is probably where your molecule gets metabolized. So essentially, you're clicking the linker away and you end up with a potent E3 binder, with a potent target binder, and you really don't know what you are looking at in your in vivo studies. And there are good examples how these metabolites can really compromise what you are seeing. For example, a very interesting study from AstraZeneca about the ER PROTACs. So this is how it looks overall. The whole set of degrader probes. You can read it in JMedChem. We also discussed covalent chemical probes in the same paper. So if you want to go a bit more into the details about covalent chemical probes, you can also go to this paper and read how we adapted the different criteria for a covalent probe. Now, the obvious question is, are there compounds fulfilling those criteria? And I will start with the kind of the older sibling in the degrader space, so the molecular glue degraders. And you can see a few examples here in this table. Again, for the experts, no surprise to see Boehringer Ingelheim's BCL6 degrader on here, comes with a matching control. So a non-grading, this should be non-degrading control. And then a number of other examples. For example, this GSPT1 degrader from the Rankovic lab. There are a few more on the horizon where data sets have been shared, and it's pretty clear that these will fulfill these criteria, but the chemical structures are just not published, and you would have to rely to some educated guesses from patents. I want to discuss two examples in this table slightly more in detail on the next slide. And I will start with Monte Rosa's GSPT1/2 degrader, which the structure is shown on this slide. And it's, I think, really impressive to see in the structure how by fine-tuning this substituent on the IMiD core, you can redesign the surface. It's almost like a point mutation and then completely change the degradation profile. Now you're degrading GSPT1 and you are not degrading any of the other well-known neosubstrates of cereblon. I think this is really impressive. You're not degrading. If you're still engaging them, I don't know. I don't have those data. If you look at Novartis Helios degrader, IKZF2, they provided such data. They also provided cereblon recruitment data showing the selectivity for IKZF2 being already apparent in the formation of the ternary complex, but there is some ternary complex formation also for IKZF1. it doesn't translate into degradation. So there's degradation selectivity, but there are additional zinc fingers which are engaged. In case of SALL4, this leads to degradation. SALL4 is hardly expressed in most cells, so this is probably not an issue to work with this compound in a target biology setup, but it's something you need to know, and it's good that Novartis put those data into the public domain so that everybody knows it. So, PROTACs. That's obviously a way more complicated question. Nobody is probably able to really follow the field about all the PROTACs which are published. You can go to some databases, but it's challenging. I've added a few more examples beyond what was listed in the JMedChem paper based on disclosures last summer. Nurix, BTK inhibitor, KGaA's IRAK4 inhibitor, and you will see the structures on the next slide, which clearly also fulfill the potency selectivity criteria. They don't come with matched controls, so that is obviously then a shortcoming. Many of the PROTACs in the public domain, especially cereblon targeting PROTACs, do not come with published selectivity data, so it makes it difficult to really assess if they would be fit for purpose. And there are also many PROTACs who are based on unspecific target binders. Yes, it's true. Take an unspecific kinase inhibitor, turning it into a PROTAC. This will lead to higher degrader selectivity, but it's still potentially an inhibitor of many kinases. If I want to study biology, this is probably not what you want to have in a chemical probe. So, as I promised, some structures. I'm starting here with the Nurix BTK case because it's a nice example to show what we as a field are now able to do. Cereblon engaging. Everybody is aware that when you're engaging cereblon, there's potential neosubstrate degradation coming for free on top. But based on what type of a cereblon engager you're choosing, the traditional thalidomide or this open phthalimide as an example, you can dial in or dial out neosubstrate activity. So when you're interested in studying BTK biology, this is obviously the way to go. Highly selective, not touching the neosubstrate. This is an excellent tool to study then what happens when you degrade BTK. There are more such examples in the public domain. They are sometimes more coming from an industry setting, which not always makes it easy for everybody to access those data. That's why I included here one example I like very much from KGaA, which was published last year at the ACS meeting, where they show how going from a pomalidomide ligand to an imidazolone, they were able to provide a highly potent cereblon engager. And this here should read micromolar. This should also read micromolar so that you don't get confused. You can see it's a highly potent engager of cereblon, very good property space, great PK, and it's completely clean on the neosubstrates. So now you can really take whatever your goal is. If you're interested in getting to really clean degraders, this is obviously a very powerful starting point. And it doesn't come as a surprise that you then find this cereblon engager also in KGaA's clinical stage IRAK4 PROTAC, the one which does not have neosubstrate activity. And there are more examples, again, BTK space from Novartis, these type of dihydrouracils, which also have been published by, for example, the Rankovic labs and by others. So there are now a number of ways how you can come up with cereblon engaging PROTACs which are very clean. This is a paper which is behind a paywall from authors from Monte Rosa and the Tomei lab, but I can really recommend this paper where they discuss a lot of the SAR and link it back to structure biology. So if you're really interested to understand more what is the state of the art of cereblon engaging, this is for sure a good paper to start with. There's probably more to come. I'm highlighting here just two papers which were uploaded this week to bioRxives back to back. We have been involved with one of them. It comes from our consortium with our friends in Frankfurt at the Goethe University, at the KNAP lab, our Merck scientists, and a number of other partners, including our colleagues at Enamine. A pretty similar study, at least with the same objective in mind and a similar solution from the Dundee lab and the Chuli lab in Dundee, both providing cereblon constructs which allow in a more easy way to screen for more new binding motifs, and this potentially then will even extend the available chemical space for cereblon engaging. Why is it important? I'm giving here one example, and I selected this because it's a really new paper, and I think the authors asking a question I find personally very interesting. And the question is, SOS1, it's a modulator of RAS biology. There has been a SOS1 inhibitor, several SOS1 inhibitor programs at Boehringer, at Bayer, with relatively, let's say, limited effects, at least reported. So what's going on? There was clearly a higher expectation what a SOS drug could potentially do. So these authors took the Boehringer SOS inhibitor and converted it into a PROTAC. It's quite active, and then did the study, which is indeed interesting, by comparing the inhibitor with the PROTAC. And they used different cell lines with a different genetic background, and indeed there are some striking differences between the two. So that's obviously a quite interesting finding. Why is it different? And then they also went to in vivo. Unfortunately, they didn't report any selectivity data. And just again, looking at the structure, there's a high likelihood that with such a attachment point, you will degrade lots of these neosubstrates. So in the absence of this information, it's really important to work with these data. There is a SOS VHL project in the public domain, and this would probably be a good tool if one would add it in the same assays to get a bit more of an understanding what's ongoing. So what I'm saying, highly relevant question. I would be very interested in the results, but there's clearly more to do to bring us then to a stage where we can create a hypothesis. Another study published very recently where I was really looking at it and smiling, saying it's showing how the community moves forward, was this one. So from the Cerner lab in I think it's in Michigan, they worked on Src kinases, published a PROTAC based on dasatinib. And first of all, what directly jumps to the eye, this is almost like a clinical stage linker when you look at it. So this is clearly something where they thought about, we want to have a really clean compound over here, and they confirmed it, published the proteomics studies, relatively high concentration, long incubation time. So they really stacked the cards against themselves to show this is a really highly selective degrader. And they even published the inhibition profile, the interactome of the kinase inhibitor. So I think this clearly shows how the field is becoming aware of what type of data do you need to then work with these compounds and start studying biology. So I was really happy to see this. It was published just a few days ago. So I talked a lot about cereblon. One important point, and I'm absolutely convinced about this, is VHL. VHL is a great system, especially if you're interested in generating probes to study biology. So with VHL, you don't have the topic with glue activity, neosubstrates. There is one reported case, but I would say in general, there's a very limited risk. There's a good understanding of the role of VHL. It's highly amenable to structure-based design. No species differences. I didn't highlight this, that this is an additional topic that cereblon, when you want to work in rodents, there are a lot of limitations. Obviously, it is very difficult to get a VHL PROTAC into an oral drug. But honestly, for target discovery studies, is this really necessary? You can dose them as C or IP, and they, in many cases, give high exposure. There's a bit of a notion that it may be easier with cereblon to actually find degraders, but I'm not sure if this is really true, and I will come back to this in a minute. Here are a few examples of published VHL-based PROTACs. Obviously, the Chuli Boehringer example, which is quite well known. there are a number of additional examples popping up in the literature. And what I really like is even for some really innovative targets which haven't been around for a long time, there are now first VHL-based PROTACs, which obviously allow to take an inhibitor and then go on to a degrader to study the biology of these still relatively, I would say, new targets like this RNA methyltransferase. Now, I mentioned that there's a bit this notion that it may be more difficult to generate a PROTAC, an active PROTAC with VHL. I think this has to do, at least to some extent, that we chemists sometimes maybe we are a bit lazy. So the beauty of the VHL system is that you have lots of different exit vectors, but the reality also is we tend to use the one which is chemically the easiest to modify, and this is over here the amide. I was told when I give a talk in the seminar, I'm expected to show at least one new BRD4 PROTAC. Otherwise, I will never get invited again. So that's what I'm doing here on the slide. So what we did, and we published this not too long ago, a study led by Johannes Krieger and Sarah Schlesinger, that you actually can even use the thiazole over here as an exit for generating PROTACs from VHL. And we generated as our BRD4 PROTACs, which are highly potent and complete degraders of BRD4, which I think emphasizes again the versatility of the system. It's a great system to do structure-based design. There are lots of ternary complexes in the public domain, and we added also a few. It seems that the induced protein-protein interaction is really specific. So you can have very different linker constructs. If you then overlay the VHL system with the same binder, in this case, even the two different bromodomains of BRD4, you see very similar induced interactions, and obviously this then opens the door for a lot of structure-based design to optimize those. It's great to see that there are many additional developments. Again, Chulilab with colleagues from Bonn publishing SAR on VHL binders, study from the Tate Lab and GSK scientists on covalent targeting of VHL will also be interesting to see what will come from these first data. So there's a lot ongoing in this space. I will summarize my talk about the degraders. Don't jump off immediately. I still have about a 5-minute teaser for another topic, but I want to summarize here at this point what I wanted to say about molecular degraders. I think they have a lot of potential, not only for sure as drugs, but also to discover new biology. So those people who discover new PROTACs, put them in the public domain, they really can influence how we further evolve our understanding of biology, but it's really important to adhere to certain recommendations, make sure that you provide clear recommendations how to use these probes, for example, by providing concentration ranges, prove what is the mode of action, provide the necessary selectivity data, be clear about what cell lines you used. I haven't discussed this in detail, that many of these data are quite specific for the cell line used, and make sure that you also put the controls in the public domain. And I think over time, as a community effort, we will be successful to convince people using these tools that they also use the controls. As I mentioned several times, so you can read what we have written in JMedChem. And while closing this part of the talk, I not only want to thank very much my co-authors. So we had very good discussions about many of these points with Joachim, Mary, Monique, and Paul. We also kind of, let's say, got on the nerves of many colleagues. So a number of colleagues from Merck who had the pleasure, I have to say, to read various versions of the paper and then contribute their thoughts. But also I bugged a number of people in the field like Alessio, Danette, Stefan, and we collected their thoughts. And I hope that they find their input and then also some thinking, some discussions we had well reflected in what we are proposing here. There is one additional, I would say, next frontier in the degrader space I'm really excited about, and I wanted to steal 5 minutes to give you a short teaser about an additional topic, which is, let's say, loosely related to what I was talking about. I've talked a lot about selectivity. I think one important question around selectivity in the degrader space is, where do I want to degrade the target? Degraders are extremely powerful drugs. You remove the target. If the target is a wild-type target, there is a relatively high risk that you see significant safety tox findings. So you want to target maybe your small molecule to the diseased cells. And what's the best way to target a small molecule to a specific cell? It's by using antibody drug conjugates. So there has been this idea to use degraders as payloads for ADCs for quite some time. And I just stole here a graphic from one of the biotechs in this field, Orum Therapeutics, where they show in a cartoon how this would work. So you have the antibody recognizes the tumor-associated antigen, gets internalized, then the antibody gets catabolized, which sets free the degrader, and then the degrader can do its magic in that cell which carries the antigen. There has been tremendous modality innovation in the ADC space with significant clinical impact now also reaching solid tumor indications. There have been now the first assets where degraders are used as payloads for ADCs in clinical testing. One is using a GSPT1 glue degrader, and then AbbVie recently put a PROTAC ADC in the clinics where it's relatively likely that it's CD33 targeting and it carries a BRD4 PROTAC. And some of you will also have seen that there have been quite significant deals recently between companies who are experts in the ADC space with companies who are experts in the TBD space, which already tells you that there's a lot of excitement in this field. Now, as a chemist, if I look at this proposal to put a PROTAC, for example, as a payload on an ADC, my immediate question is, oh, this sounds like complex chemistry. I'm showing you here now some graphics which I stole from an excellent review, probably the expert in the field, Genentech's Pete Dragovich, who describes the general setup of these ADCs, where you have the drug and then you have a certain, let's say, mechanism how you want to set the drug free, and you encode this mechanism in the linker. So what does it mean for a PROTAC? So going back to a BRD4 PROTAC, this time it's Genentech's own VHL BRD4 PROTAC. How would you now put this on an antibody? What they used is a carbonate linker, essentially using the critical hydroxy and then linked it via a disulfide to an antibody, which essentially means it gets catabolized and then it's self-cleaving to free up the PROTAC. Another example, and that's probably addressing a bit the question about the stability of this carbonate, what you do in many of these ADCs, or what is a typical attachment point for the linker, and then the antibody is a primary amine. However, then you have to redesign your linker to have your attachment point, which they also did, and then they used an enzyme cleavable linker to get the full ADC. And I think it's clear to go from here to here. That is quite some chemistry muscle you need to apply to get something like this working. Now, wouldn't it be cool if you just take the free PROTAC and this would be directly recognized by the antibody? No chemistry. So that is the question. There was a very talented, there still is a very talented team at Merck who had also this idea, could we generate antibody PROTAC conjugates without the need of doing linker chemistry? And the idea to do this was to grow heavy chain only antibodies, so nanobodies, who are able to recognize, to bind the PROTAC. So that's essentially what they did, grow these nanobodies who are able to recognize the PROTAC and then genetically fuse these nanobodies to the C terminus of therapeutic antibodies. And these domains, they termed then monovalent immobilizers of chimeras mix to generate something which is now a non-covalent antibody PROTAC complex. And this is how it looks in reality. So you take your antibody, you take your PROTAC, essentially you pipette it in at room temperature. And what is shown here is the size exclusion chromatography native MS. And you can beautifully see if you do this with a stoichiometry of 2:1, you end up with almost a perfect distribution. I think mathematically it's 1.9. You can read the details in this bioarchives paper of the loaded antibody PROTAC shuttle. We call it the PROXEP shuttle. So the PROTAC meets the antibody. And here's an ITC data essentially telling you the same nanomolar affinity, about a 2:1 ratio, as you would expect if you titrate it this way. Here's a bit of chemistry or SAR. What was used is exactly the same PROTAC I had shown you previously. So this BRD4 PROTAC, which was published by Genentech, highly below nanomolar affinity of the MIC, which was selected here. And then they looked at some variations of the VHL part. And it's interesting, again, for more the people who usually design PROTACs, it's accepted that there is a magic method. You can also have the rather typical oxygen here on this part of the molecule. You still have high affinity, so you can really capture a lot of PROTACs which have this traditionally used VHL binder. Where the affinity takes a hit is when you turn around the oxygen, the hydroxy group, which is typical for VHL binding. Now, what can you do with these molecules? Here are some cellular studies. Let's maybe start on the bottom left, A431. This is an EGFR expressing cell line. If you treat it with a free PROTAC, this is a red dot, you effectively kill those cells. If you now use the PROXEP shuttle, which is generated from an EGFR recognizing antibody with the MIC7 nanobody, which is able to bind the PROTAC, you see more or less the same cell killing curve. If you use a non-matching control where you are not binding the EGFR on the surface, you effectively detoxify the PROTAC. And you can see the same if you do this with the CD33 PROXEP shuttle. Here, the number of receptors on the surface is lower. That's why you have this kind of this in-between setup. But obviously, and I should point this out, this is a construct where the PROTAC antibody ratio is 1. And you can already guess, looking at the data, and I mentioned the effect of the number of receptors on the cell surface, how you can now design or modify what you're doing to broadly opening up this window. And if you go to the receptor negative cells, you see exactly what you would expect to see, that you detoxify with both shuttles while the PROTAC itself kills the cells. So that's what I wanted to share for this. I think it's a powerful new technology. We call it, as mentioned, the PROXEP shuttle, where you are able to generate non-covalent complexes between an antibody and a VHL-based PROTAC and deliver them to cells recognized by the antibody. I've shown in vitro data just as a teaser to make sure that you really go to this paper. You will also find in vivo data. So this also works in exposure studies and xenograft studies. So there's, I think, a lot of potential. If you're planning to come to the AACR annual meeting in April to San Diego, come early so that you are there for the educational session, because you will probably hear more about this technology. So, second part of my acknowledgement. Great team of highly innovative and engaged scientists who turned this idea into reality. This is all I wanted to share today. So some recommendations around quality criteria for probes, but I also wanted to show you this exciting new technology to deliver PROTACs to cells. And here I stop, and I hope, despite the, let's say, delay in the beginning, there are still a few minutes left to answer some questions.